Hello, I'm Hyun Lee from the Biomedical Science Department of SNU. I'm studying microbiology and immunology in Bum-Joon Kim's lab. Thank you for giving the opportunity to present my research. Today I'm going to talk about the heterologous effect of live MPG in NK cells mediated by crosstalk between disease and NK cells. I'd like to give some background on my research before we get started. It is known that live vaccines are associated with very beneficial non-specific effect called the heterologous vaccine effect. There is strong clinical evidence that suggests the live vaccines can reduce all-cause mortality from various infections. The concept of trained immunity has been introduced to explain heterologous effects of certain live vaccines that is mainly mediated by monocytes or NK cells. Although the term trained immunity was not used in this manuscript, this study started from the concept of trained immunity because it is intimately related to the heterologous effect. Our research group has studied a temperature-sensitive mycobacterium which is called MPG, and we reported its immune induction ability against tuberculosis and Mycobacterium sepsis. After the study, we found that live MPG vaccination promotes interferon gamma production against C. albicans and a robust production of interferon beta in disease in response to live MPG infection. We know that our bodies make interferons as a part of our immune response to many microbial agents, and interferon binding to the receptors on the cells that signal being translated to the nucleus turns on many genes, on or off, and many of those have to do with the immune system. For that reason, in some microbial infection models, NK cells are activated by DC-derived type 1 interferon and inflammatory cytokines. And this DC-derived type 1 interferons manage crosstalk between disease and NK cells, which enhances NK cell activation and cytotoxicity. Since type 1 interferon response can be activated by sensing viability-associated PAMP in terms of VitaPAMP, which serves as signal instigators involved in the recognition of microbial mRNAs or cyclidide nucleotides in host cells. In this study, we aim to explain heterologous immunity of live MPG based on DC and NK cell crosstalk and elucidate the mechanism underlying type 1 interferon response in disease in response to live MPG infection. This is our workflow. We first demonstrated heterologous vaccine effect of live MPG by NK cell activation and compared type 1 interferon responses between live MPG and heat-killed MPG-infected disease. And we also needed to make sure if DC-derived type 1 interferon plays a direct role in activating NK cells in response to live MPG infection. And we try to find out what is not just the activation but also the functionality of these NK cells that might be important for getting them to work. And we also evaluated heterologous immunity of live MPG in human NK cells. First of all, to assess the potential of live MPG to induce heterologous immunity, we isolated NK cells from vaccinated mice and re-stimulated them with unrelated pathogens. And we found that the live MPG group showed robust interferon gamma and TNF-alpha production, indicating that NK cells will be a major source of the enhanced interferon gamma level. Next, we evaluated the activation levels of NK cells in response to live MPG infection. We analyzed the levels of NK cell activation in spleen, blood, and liver tissues. I brought only results obtained from the spleen in this slide. We found that both the frequency and number of activated NK cells were increased in the live MPG group compared to other groups, with no significant differences in the total NK cell subset between the vaccinated group. To further determine whether these effects could lead to actual non-specific protective effects in vivo, we vaccinated skin mice to exclude the involvement of adaptive immune system. After infection with a lethal dose of C. albicans, we found a significantly lower fungal burden in live MPG vaccinated group, indicating that this non-specific protective effect was induced based on the innate immune system. And notably, we've identified NK cells from the live MPG group showed enhanced cytokine production, supporting that the non-specific protective effect of live MPG against C. albicans appears mainly due to activated NK cells. Based on these findings, we investigated whether live MPG induces enhanced cytokine production in disease that mediates NK cell activation. We tested several mycobacterial strains to compare the production of interferon beta and found that live MPG elicited a strong interferon beta response compared to any other strains. And we also detected enhanced IL-12 and 15 production in live MPG-infected disease. Following this observation, we identified an increased level of phosphorylated IR3, which is an essential transcription factor involved in the induction of type 1 interferon gene expression. Since it is known that type 1 interferon response is associated with the ear stress response during microbial infection, we investigated related ear stress molecules as the upstream signal of type 1 interferon response. Indeed, we found that live MPG activates IR1 alpha, which is turn led to XBP1 mRNA splicing, whereas neither BCG nor he-killed MPG does not. In addition, since the IR1 alpha mediated unfolded protein response activates the type 1 interferon response via TBK1 activation, we also identified activation of TBK1 in live MPG-infected disease. So far, we have found that live MPG showed the opposite type 1 interferon response to he-killed MPG, and now we are going to find out why. We first measured the levels of different cyclic dinucleotides from live MPG as VitaPAMP by HPLC and identified that the level of CDI-GMP was higher than within other strains. Furthermore, since our in silico analysis of live MPG genomes showed that 9 GGDEF yield domain proteins were involved in CDI-GMP metabolism, we hypothesized CDI-GMP could act as a VitaPAMP. To investigate whether CDI-GMP serves as an instigator of type 1 interferon responses in live MPG-infected disease, we cultured mycobacterial strains with sulfathiazole, which is known to interfere with CDI-GMP biosynthesis. As expected, we found that interferon beta production was significantly reduced in disease when sulfathiazole-treated live MPG was used for infection. Since it is well established that CDI-GMP binds directly to sting, which is an innate immune sensor linked to ear stress response, we verified the sting dependency of live MPG-induced type 1 interferon response by treatment of antagonist of sting, H151. Interferon beta production was reduced by H151 in a dose-dependent manner in only live MPG-infected disease. On the other hand, in live MPG-infected disease, we found an increase of cytosolic cGAMP, which is synthesized by cGAS via a combination of CDI-AMP and GMP, and is also considered to be a type 1 interferon stimulator by binding to sting directly. So we treated an inhibitor of cGAS RU521 to determine whether type 1 interferon response changes as cGAMP synthesis is disrupted. And we found that RU521 treatment also led to a reduction in interferon beta secretion in live MPG-infected disease. To verify the connections between type 1 interferon production induced by CDI-GMP or cGAMP of live MPG and ear stress responses, we compared the induction of ear stress responses with or without sulfathiazole treatment. We found that a decreased ear stress response led to reduced activation of TBK1 and IR3 in the cells infected with CDI-GMP depleted MPG. And reduced LC3B levels were also detected after sulfathiazole treatment. These results suggest that CDI-GMP is a VitaPAMP of live MPG, inducing an ear stress-mediated type 1 interferon responses in a sting of or cGAS sting-dependent manner. Next, we examine the direct role of disease in activating NK cells in response to live MPG infection. We co-cultured naive NK cells with infected BM disease and evaluated the activation levels and heterologous immune responses of NK cells. We identified NK cells were activated by live MPG-infected disease and enhanced cytokine production after restimulation with unrelated pathogens when NK cells were co-cultured with live MPG-infected disease. These results showed that live MPG can induce disease-mediated NK cell activation and heterologous immunity. Furthermore, we try to verify the impact of impaired interferon beta signaling on NK cells by using BMDCs from type 1 interferon receptor knockout mouse. In response to live MPG infection, we confirmed that defective interferon beta signaling in BMDCs led to reduced NK cell activation and heterologous immune responses. We further evaluated the functionality of these NK cells if the non-specific protective effect of live MPG vaccination is enhanced by NK cell-mediated cytotoxicity. We prepared BMDMs infected with unrelated pathogens as target cells to assess the cytotoxicity of NK cells from vaccinated mice. By confirming the high cytotoxicity of NK cells in the live MPG vaccinated group to infected cells, we could ascertain that live MPG stimulated NK cells mediate protective effects through enhanced cytolytic activity. The expression levels of granzyme B and perforin 1 were also highly elevated in NK cells from live MPG vaccinated mice compared to other groups. Lastly, we performed functional investigations by using human PBMCs to see if live MPG can also induce heterologous immunity in human NK cells. Consistent with our observations, we detected increased interferon beta production in only live MPG-infected human disease, depending on the presence of CDIGMP. And we also identified the enhanced activation levels and heterologous immune responses of NK cell among live MPG-infected PBMCs. We also found that live MPG-stimulated NK cells show increased cytotoxic activity against infected human macrophages with upregulation of cytotoxic proteins. In summary, we show that live MPG but not dead MPG enhances heterologous immunity against unrelated pathogens in NK cells by interferon beta of disease. CDIGMP from live MPG acted as a vitapamp, leading to sting-dependent type 1 interferon production in disease via the IRE1 alpha and XBP1 pathway. We also demonstrated that increased cytosolic cGAMP by cGAS can induce type 1 interferon response in disease by live MPG infection. We found that DC-derived type 1 interferon plays a pivotal role in NK cell activation in response to live MPG infection, showing NK cell-mediated non-specific protective effects against C. albicans infection in a mouse model. Our findings not just demonstrate the crosstalk between disease and NK cells, but also explain the molecular details regarding heterologous immunity in NK cells following sensing vitapamp by disease during live mycobacterial infections. And this is all about my presentation. Thank you for listening.